Bismillah ar-Rahman Rahim. We start another November 23 exam. This is the question paper 2-1, and let's see how many questions I can handle in this. And please do subscribe and do leave comments as how I can improve these videos and how I can help you more. And thank you very much for watching. Now, starting with question number 1, it says scientists measured the concentration of sodium ions and potassium ions in the red blood cell and in the blood plasma of a group of people. The results are shown in table 1.1. So red blood cell sodium ions were 10 and potassium ions were 100, and in the plasma they were 100, and in the plasma the potassium ions were 4. Now it says use the information in table 1.1. So always look at this table. You don't talk something about something else which you know, but look at the table and then give me some information. Identify and describe the process by which potassium ions enter red blood cells. So potassium ions enter red blood cells. Now where are the potassium ions? Concentration of potassium ion in the red blood cell was 100. So this is the red blood cell, and inside it was 100, and outside in the plasma it was 4. So always do this little diagram. You can write on the paper. Who says you can't write on the paper? So inside the red blood cell it was 100, and the plasma it was 4. And the sodium ions, what was in the sodium ions? The sodium ions were inside the red blood cells were 10, and in the plasma they were 100. So you see how they are trying to check whether you know something or not. So how do the potassium ions, now we're talking of the potassium ions. So this is the potassium ion story. So they were 4 and they were 100. So how would they enter? They would enter by active transport. It's very easy to get the 3 marks because you just have to give me the whole definition of active transport. Active transport against the concentration gradient using energy, or you could have said using ATP, and this results in a conformational change of the protein carrier. And these protein carriers would have specific binding sites. Number 1 for the ion, and for the ATP must be binding to it as well. So you had to write it. You give me more points so that you get your marks. This was something in which nobody should have lost any marks in this question because it was very, very basic. It was very easy. So active transport against the concentration gradient. You gave me the figures from 4 to 100. You gave me that using energy, or you could have said using ATP, because ATP is the universal energy currency. And then there is a conformational change. The conformational means that there's shape change. If you don't know it, just Google it. What does conformational change mean? That's how you increase your vocabulary. Then it says part 2 of the question, sodium ions and oxygen molecule enter red blood cells. State one similarity and one difference between the processes used by sodium ions and oxygen molecules to enter red blood cells. One similarity and one difference. Similarity was what? Diffusion down the concentration gradient. It's a passive process. Diffusion down the concentration gradient is a passive process and does not require any energy or ATP. The difference is that one is an ion and one is a small uncharged molecule. So sodium ions will enter by facilitated diffusion and oxygen molecules will enter by simple diffusion. Simple and facilitated diffusion, I hope you know the difference. Simple diffusion is through the phospholipid bilayer. Facilitated diffusion is through a channel protein in the phospholipid bilayer. So the sodium ion will use a carrier protein, or you can say a channel protein, or you can say they do not pass through the phospholipid bilayer because there's the hydrophobic part of the bilayer which will not allow ions to pass. So similarity will be that both are a form of diffusion, both are passive processes and no energy or ATP is needed. Difference will be sodium ions enter by facilitated diffusion and oxygen molecules enter by simple diffusion. Or you can say sodium ions use a channel protein because they cannot pass through the hydrophobic part of the bilayer. Part 3. Chloride ions move across the membrane of human red blood cells in a process called the chloride shift. Explain why the chloride shift is important for the transport of carbon dioxide from respiring tissues. Now, either you know this or you need to go back and revise this if you don't know this. Hydrogen carbonate ions move out of the red blood cells and chloride ions move in. That is the chloride shift. Hydrogen carbonate ions, HCO3 negative ions move out of the red blood cell and chloride ions move in. Now, why is that? Why does that happen? To maintain electrical neutrality inside the cytoplasm of the red blood cell. To maintain electrical neutrality, because you see, if so many negative ions move out and the positive ions don't move out, so the red blood cell will become positively charged and they will repel each other. So that should not happen. So you could have said to maintain an equilibrium to increase the reaction between the carbon dioxide and water. So you have to be knowing this point of the syllabus. I mean, I'm not going to be revising the syllabus here. You need to revise this part of the syllabus if you're not very clear on this. So you must know how to word this. HCO3 negative ions move out of red blood cell and chloride ions move in to maintain electrical neutrality. Or you could have said to maintain the equilibrium to increase the reaction between carbon dioxide and water. Now this is 2 marks. It doesn't mean you have to write all these 3 points. Any 2 of these would get you the 2 out of 2. Part B, scientists study the uptake of a substance F by the human red blood cell. The red blood cells were immersed in a solution of substance F for 30 minutes. After this time, the scientists recorded 2 observations. The cell surface membrane of the red blood cell showed infoldings, invagination, an increase in the number of vesicles in the cytoplasm. Identify the process. So you see, if the red blood cell is going to invaginate, and this will be what? Endocytosis. So endocytosis or pinocytosis or even bulk transport was allowed. Question number 2, figure 2.1 is a transmission electron micrograph showing a section of a specialized epithelial cell found in the lining of the stomach. It's not in the syllabus, remember that. So it's something they're going to test which is unknown. This cell produces extracellular proteins, proteins that are working outside, like pepsin, the enzyme, that are released into the bloodstream. So showing a section specialized in the cell produce extracellular proteins that are released into the bloodstream. So it makes these cells which are then thrown out and transported in the blood. Now you can see there's X and Y. Now you must be able to read micrographs. So we have this micrograph. It says outline the role of structure X. Now you can see this is the nucleus. This appearance of the nucleus is very, very typical. And inside the nucleus is this darkly staining area. So outline the role of it. They're not asking you the role of structure X and structure Y. Structure Y is, of course, the chromatin or the chromosomes or the DNA as shown in figure 2.1 and the production of extracellular protein. So inside the nucleus, you would have the chromosomes, which you are not, of course, seeing. So structure X is the nucleolus. So what does it do? It synthesizes ribosomes. What does Y do? The chromatin. It contains genes coding for the polypeptide, for the proteins, or it is the site for the transcription of mRNA. So the nucleolus synthesizes, you could have said ribosomal subunits, you could have said ribosomes, both were correct. Or you could have said synthesis of organelles that are the site of protein synthesis. You could have said that as well. The Y was a chromatin which contains genes which code for that protein or that polypeptide. And it is also, you could have said, site of transcription where the mRNA is produced. Part B says the cell in figure 2.1 releases ghrelin, a small protein that acts as a cell signaling molecule. Figure 2.2 shows the sequence of amino acids. What is the sequence of amino acids? That's a primary structure, the number and sequence of amino acids in a ghrelin molecule. The amino acid serine in position 3 in figure 2.2 has been modified by the addition of a saturated fatty acid chain. Saturated fatty acid chain. Right? Everybody clear? It has 28 amino acids. State the level of protein structure shown, primary. Anything showing the number and sequence of amino acids will be the primary structure. If it shows hydrogen bonds showing the alpha helix and the beta pleated sheet, that would be the secondary structure. Then you say state one similarity between the structure of a saturated fatty acid molecule and an amino acid molecule. Well, they both have a carboxylic acid group and they both contain carbon, hydrogen, and oxygen. So contains carboxylic acid group, and you could have either said this or you could have said contains carbon, hydrogen, and oxygen. Part 3 says the addition of the fatty acid chain allows ghrelin to function as a cell signaling molecule. Suggest how the addition of this fatty acid chain allows a ghrelin molecule to act as a cell signaling molecule. Now, first of all, you must know what is a cell signaling molecule. You see, why would insulin work on the liver cells? Now, insulin works on the liver cells because this is the liver. In the liver, the cells have receptors. So the insulin molecule goes and fits onto it. Cell signaling molecules would have receptors on the target organs, and there the ligand or the cell signaling molecule will work. So the main thing you've got to decide is this will give it an overall 3D shape, allows the ghrelin to bind to its receptor. Ghrelin is to have a complementary shape because it's a ligand, it's a signal, so it has to bind to the, it's complementary to the receptor. If the receptor is like this, then it's just like the active site story, you see. But we don't talk of the active site here. So the shape of the ghrelin molecule will be complementary to the receptor. Interacts with the hydrophobic region of the receptor molecule. Or you can say helps in embedding in the hydrophobic part of the cell surface membrane. So it was very basic and easy. You only had to give me 3 points, only 3 marks for it. So you could have generally termed it if you knew the cell signaling molecule and the fact that the shape of it is complementary to the receptor, then you could have come up with all those points and not get very confused and write something which is totally rubbish. So forms part of the overall 3D shape. The fatty acid will become part of the overall 3D shape, and then this will allow the ghrelin to bind to the receptor or the part of the ghrelin to have a complementary shape to the receptor. And then you could have also said getting it is going to interact with the hydrophobic region of the receptor molecules because the fatty acids are in the whole hydrophobic part of the phospholipid bilayer. So you could also have given me that point, but if you really understood it, then you'd be able to give me this point. Scientists have discovered that the gene coding for ghrelin contains 5,000 base pairs. ATGC, ATGC. So this is 3 base pairs. Now that is 5,000 base pairs. This is a much larger number of base pairs than is needed to code for the ghrelin molecule shown in figure 2.2. Calculate the percentage of base pairs found in the gene that codes for the ghrelin molecule shown in figure 2.2. Show your working and give your answer to 1 decimal places. Now, why did we do that? Codes for 29 amino acids and a stop codon. So 30 amino acids into 3. 30 into 3 would be how many? Divided by 5,000 into 100. This is equal to 1.8%. Because you see, if you see in the molecule is 29 codons for 29 amino acids and a stop codon. So 29 and then 30, 30 into 3, each amino acid is coded by 3 bases. You see, 3 bases code for 1 amino acid, AGA or ATA or AAC. So 30 into 3 divided by 5,000, and this would be 1.8%. Then part 2 of the question, the primary transcript produced from the ghrelin gene is a longer molecule than the mRNA found in the cytoplasm. Explain how the primary transcript is modified before translation. Basically, we all know that introns removed, exons are joined together. This is called RNA splicing. So you have to say introns removed, exons joined together, gene splicing or RNA. Please do not ever write DNA splicing. This is not DNA splicing. That's a reject. That means a large number of students must have written that they had wrote DNA splicing. So you see, DNA from DNA we get pre-mRNA, and then from pre-mRNA we get mRNA. Question number 3. Plants have specialized cells for the efficient transport of assimilates. Table 3.1 shows the features of two different types of cell found in plant tissue, cell type A and cell type B, adapted for the efficient transport of assimilates. Cytoplasm, cell type A, both of them have it. Nucleus is not present in cell A, present in B. Mitochondria, few, many. Cellulose in the cell wall present in both. Ribosomes not present in A but present in B. Many plasmodesmata connect type A cell with type B cell. Well, if it has many mitochondria, it has to be companion cell. Identify cell type A and explain why the plasmodesmata are important to cell type A explanation. What is the explanation? A is a phloem sieve tube element, and it connects the cytoplasm of the two cells. So this will allow rapid diffusion of sucrose from one cell to an adjacent cell, and this will result in the efficiency of loading and unloading from the sieve tubes. You see, this is the sieve tube. You have to understand these are the sieve tubes and these are the sieve plates, and next to it is a companion cell, and the companion cell has a lot of plasmodesmata in it. It has these gaps. So when the sucrose is loaded into here, then it's going to flow through here and easily go into the phloem sieve tube element. So the phloem sieve tube element has to then transport this, the assimilates. Assimilates means what? Sucrose, amino acids, which the plant has made, which has to be transported from the source to the sink. Source to the sink. So cell type A is phloem sieve tube, connects cytoplasm of the two cells, rapid diffusion of sucrose, efficiency of loading and unloading from the sieve tubes. B part says the polysaccharide callose is found in the cell walls of cells close to plasmodesmata. Figure 3.1 is a simplified diagram shows the structure of callose. Not all hydrogen atoms are shown. Compare. Now this is something which is a new thing comes and then you have to do it and you get very confused. This is very typical of you. Compare table 3.2. Compare the structure of a callose molecule which is given here with a cellulose molecule. Now you know cellulose is made up of beta glucose. What do you see here? This is also beta is what? Alpha below, beta above on carbon 1. Alpha below, beta above. Now you look at it very carefully. Now these of course also have on carbon 1 they are above. So this is also beta glucose. Then bond connecting the monomer is 1,4 glycosidic bond in cellulose. Here what you see is 1 and 3. Here it's 1 and 3. This is the glycosidic bond. So this is 1,3 glycosidic bond. Now the shape of the molecule is helix. This is not helix. This is straight or linear. Or you could have said linear. Someone will say linear. Then orientation of the glucose, alternate glucose are rotated by 180 in cellulose. Here they are the same way up. So adjacent glucose molecules same way up. They are not rotated 180 degree like cellulose. Then the part C says hydrogen bonding is important in the movement of water through xylem vessels in a plant. Describe the role of hydrogen bonding in the movement of water through the xylem vessels. So hydrogen molecules give adhesion between water molecules and cellulose. Hydrogen bonding gives cohesion between water molecules. The formation of continuous columns, transpiration pull, and water evaporating from the spongy mesophyll cells creates a tension. So you have to give me all these points to understand, but don't just say cohesion. Cohesion is not getting any marks. You tell me a little bit more about cohesion. Cohesion between water molecules. So hydrogen bonds, they are hydrogen bonds. But what do the hydrogen bonds do? When they are between water molecules, it's called cohesive forces or cohesion. When the water molecule bonds or binds or attaches to the cellulose cell wall in the xylem vessel wall, then that is called adhesion. It's just like mountain climbing, and you catch onto these rungs if there are these on this when we do these at these adventure lands. So you got these things sticking out and you catch onto them and climb onto the wall. So cohesion between water molecules, hydrogen bonding gives cohesion between water molecules. Hydrogen bonding gives adhesion between water molecule and cellulose. Transpiration pull. Now, why is there transpiration pull? Because the water evaporates from the spongy mesophyll cell surfaces, and that results in a tension, and that results in the water being pulled up. So the wordings: adhesion between water molecules and cellulose. Hydrogen bonding gives cohesion between water molecules, forms a continuous column of water. Transpiration pull, evaporation of water from the spongy mesophyll cells, and water vapor lost from the leaf. Debarbed water that has traveled through xylem vessel reaches the leaves. Cooling of the leaf occurs as a result of the evaporation of water during transpiration. Water has a high latent heat of vaporization because water molecules form hydrogen bonds. With reference to hydrogen bonding, suggest why cooling of the leaf occurs as a result of evaporation of water during transpiration. You see, hydrogen bonds between the water molecules must break for the water to change its state from a liquid state to a gaseous state, from liquid water to water vapor. So the hydrogen bonds must break. Now this results in the loss of heat or energies needed for evaporation. So this will cool the plant. This happens in us as well. When we produce sweat, it doesn't cool us down, but the evaporation of sweat cools us down. So this is the O-level kids. When we enter this room is very hot. We start sweating. But the production of sweat doesn't cool you down. It is the evaporation of sweat which is going to take body heat, and that's going to cool you down. So hydrogen bond between water molecules must break for water to change state, that is convert from a liquid state to a gaseous state, or liquid water to water vapor. And the second point, because there are 2 marks, the second point would be the loss of heat for evaporation will cool the leaf down, because the plant will lose heat, will give up some of its heat. That's how we cool down as well. So question 1 to question 3 answered in this video. The rest will continue in the next video.